Good afternoon, students. Today, this is the last chapter of this course this semester, and so I've uploaded this chapter as learning activities between the dates 11th May to 17th May, which is the last week. So this is a very small chapter, and each slide is informative and it's very important. So I hope you will pay attention to the information. This is less of conceptual, but more of information. So I'm sure you know that we have 46 chromosomes, which are sets of two sets of 23 chromosomes, 23 coming from each parent. And these chromosomes are made up of DNA and proteins. And DNA part is composed of 3,100 mega base pairs. So a kilo base pairs would be 3 zeros. After 3,100, the mega base pair will be 6 zeros after 3,100. So that's a 3,100 mega base pairs for haploid genome, that is 23. And 6,200 mega base pairs for diploid genome. And out of this, 2 to 3% code for proteins or express RNA. Rest of the genes are mainly regulatory. So through various these information are obtained through bioinformatics analysis, and so there are different websites, and they report different data, but they are not that different from each other. So if you notice that the protein coding genes by gen code is 19,901, the LINC RNA genes means long non-coding genes, long non-coding RNA genes. So a gene could transcribe, and then that RNA could be translated, but there are some genes which can only transcribe but may not be translated. That's what so far the technology goes up till now. Then antisense RNA, miscellaneous RNA, pseudogenes. So total transcripts in our cell is at a given point could be 203 or almost 204,000, and so this data has been analyzed by various groups, which represents various websites. So this represents the number of genes per chromosome, right, going from 1 to 23 per haploid genome. Yes. And then these genes, using various tools, can be classified into enzymes, membrane proteins, signal transduction proteins, RNA transcriptional protein, cytoskeleton. So this is a pie chart that gives the percentage contribution of various proteins. Some of the important proteins are BRCA2, which is located on 13 chromosome, the cystic fibrosis gene on chromosome number 7, GAPD8, which is a housekeeping genes, that is glyceraldehyde 3-phosphate dehydrogenase. So let's see, where did the story of DNA started? So the story of DNA started in 1870 when Michel discovered some chemicals, acidic chemical in the nucleus, which he referred as DNA, 1870. In 1940, Avery proposed DNA as a genetic material. Then 1953, Watson and Crick discovered structure of DNA. So this is the history of DNA. In 1983, polymerase chain was discovered. In 1920, the Human Genome Project began. And so in 2009, next generation sequencing of human genome studies begin. And in 2000, so you can see that how the field of molecular biology has progressed over the course of year, beginning from mid 1800s. And all these important concepts have been translated into technologies. And this technology is used to improve human health and society. And as the knowledge increased, the sophistication of DNA sequencing also increased. And so this is more detail that you can look at later about the history of human genome. So the mapping strategies has been various, but what has happened that human genome DNA has been fragmented, and then they have been cloned into back library, and this back library has been sequenced, and then these sequences have been assembled. So what is a back library? A back library is bacterial artificial chromosome that acts as a vector to artificial carry DNA into the cell of bacteria such as E. coli. And in general, back clones can carry inserts of 200,000 base pairs. Recently, back has been used to create back libraries for preferable genome sequencing, physical maps, so on and so forth. So there are two categories of DNA markers, type 1 and type 2 marker. Type 1 mainly refers to the protein coding genes. They are slightly polymorphic. Type 2 refers to non-polymorphic, non-protein coding genes, which are highly polymorphic, which can vary from individual to individual. It can vary from species to species. So this can be tracked to understand evolution of certain species, including humans. Then there's a large portion of the DNA within our genome that is very repetitive. This could be tandem repeats, and these tandem repeats could be classified as rDNA and satellite DNA. These satellite DNA could be of three types: microsatellite, minisatellite, and macrosatellite. This is very important. So a microsatellite is short tandem repeats that consist of 1 to 9 base pairs, rich in A and T. They are also called as simple sequence repeat. Whereas my mini satellites, they are tandem repeats. They are rich in G and C. So major difference. Microsatellite, they are rich in A and T, whereas mini satellites, they are rich in G and C, and they are called as variable tandem repeats. And macro satellites, so these mini satellites, they are 2 to 5 base pairs, overall length of 70 to 200 base pairs. They are most variable in copy numbers among people. Microsatellites, they are near telomeres. And macrosatellites, they are both near centromeres and telomeres. So these knowledge of human genome has been used to interpret various individuals to understand their mainly disease process or evolution process. So this is a very small chapter, and I finish with this chapter, this course on molecular biotechnology for this semester.